Hi everyone. I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past and present and the elders from other communities whom may be here today. And today, our second last postgraduate lecture on hallmarks of cancer. It's my pleasure to introduce Associate Professor Clare Scott to speak on the role of DNA repair in cancer. Clare is a clinician scientist. That means, as well as heading a lab at WEHI, she's a medical oncologist in breast and cancer and ovarian cancer at the Royal Melbourne and Royal Women's Hospitals. Clare is the recipient of several awards, and I'll just mention the Burnett Prize in 2009, the Corey Leadership Fellowship in 2010, and in 2012, the Sir Edward Dunlop Research Fellowship from the Cancer Council of Victoria. And a little bit of her career after her medical degree at the University of Melbourne. Clare did a PhD at WEHI in Glenn Begley's lab, and then she returned to the clinic, believed to do her specialty in medical oncology. She then did postdocs in Andreas Strasser's lab and then at Cold Spring Harbour in Scott Lowe's lab. In 2010, she returned to WEHI, started her own lab, focusing on ovarian cancer, particularly difficult cancer to treat, and she'll tell you about her interest in how many cancers have faulty DNA repair mechanisms and how that pathway might be targeted. Thanks, Clare.
Thank you. Thank you. It's a real pleasure to be here today, and what I hope you'll take away from this talk is the fact that unravelling DNA repair, when we look back in the future on now, we'll think of it a bit like we now understand much more about oncogenes and apoptosis, particularly in this forum here. These are my disclosures, and relevant for today particularly are AstraZeneca and Clovis Oncology, as they both are attempting to bring Parp inhibitors to the market. So DNA repair is a therapeutic target, and we want to harness it to improve the success of current and novel treatments. In particular, we want to look at therapeutic approaches that can be designed to exploit DNA repair defects, but also, really importantly, use these drugs and our models to understand how to circumvent resistance to PARP inhibitor to DNA repair inhibitor therapies in the clinic. Getting a drug into the clinic is only the first part of the process. We then need to understand how to make that drug work better. I will discuss the concept of super responders, which are patients who have incredible responses to treatments, but in the main, that is not the case, and that's why we need to do more research. So much of my talk will focus on the early discovery of this class of drugs in ovarian cancer, but I'll also describe a little bit how this is relevant for a wide range of cancers. So why is DNA repair important? You've been focusing on hallmarks of cancer, and that's where I'm going to start and end. These are the descriptions that you'd be used to for these hallmarks: sustaining proliferation, resisting cell death, evading growth suppressors, activating invasion and metastasis mechanisms, inducing angiogenesis, and enabling replicative immortality. But throughout my talk, I'm going to refer to these hallmarks in slightly different ways as well. So in order to escape from drug control particularly, cancer cells need to harness some of their very best activities. And not all of the cancer cells do all of these, but these are the things at their disposal. They're out of control, they won't die, they won't listen, they won't sit still, they want to live forever. And it's all about me. They harness all of the energetic blood supply that's around. So that might make you think of someone young that you know, but bottom line is cancer drugs need to attack these mechanisms. So we need to be able to starve cancer cells. We need to be able to control them, stop them spreading, make them age faster, cut off their supplies, and ultimately kill them in a useful time frame. We thought we were getting on top of it, and then there were more. So the next generation, deregulation of cellular energetics, the immune system, both of which are emerging hallmarks, and then these two enabling characteristics: genome instability mutation and tumour-promoting inflammation. These are distinct from the 6 hallmarks of cancer. So the hallmarks are distinctive and complementary capabilities that enable tumour growth and metastatic dissemination. But these enabling characteristics are thought to be crucial to the acquisition of the hallmark capabilities. So that really means these are the only two you need to know in great detail because they underpin everything else. And then there are the two emerging hallmark capabilities, and the thing about them is that they really involve the tumour microenvironment. So these are newer areas that have become much more well understood in the last 5 years, but are not generally as well understood as the previous 6. So the concept of enabling characteristics being crucial to the acquisition of hallmark capabilities, we're going to focus today on genome instability and mutation, but you'll see that there is a common theme, and that's where I'll end up with at the end, around tumour-promoting inflammation. So with genome instability and mutation, this has been shown to be causally associated with the acquisition of these other hallmark capabilities, and it's best reflected by this sentence from the very famous hallmark update paper. Defects in genome maintenance and repair are selectively advantageous, that's the holy grail of a cancer cell, and instrumental for tumour progression, if only because they accelerate the rate at which evolving pre-malignant cells can accumulate favourable genotypes. So again, this concept of favourable genotypes, meaning something that is Darwinian, it will be selected for, and it will evade whatever you attempt to do to it. So what are the therapeutic applications of these, and how can we make a difference for patients? Because that's really why we're all here. So bottom line is we have to evolve our therapies faster than the cancer cell can evolve to evade them. Sounds simple, but at least if we know that is what we're aiming for, we'll know what we need to be studying, which has not always been the case. So again, Hanahan and Weinberg offered this very straightforward concept of drugs, different types of drugs, how they all impacted. And you can see some of our absolute favourites over here, BH mimetics, tyrosine kinase inhibitors. We know that the CDK inhibitors tie in very closely to these, and in the future I'll consider those as one group. My bias down here, Parp inhibitors, and I think for WEHI particularly, a lot about inflammation and the immune system. So you can actually look at these concepts and decide how and for which cancer each of these is likely to be relevant. And this is what we're trying to do in each case. So these phrases are really putting the imperative on us to be understanding which cancer cells are using which mechanism and what we need to do about it. We actually have drugs for all of these. Some will be names known well to you, some indeed very old names, some almost branded names which are less familiar to us than ABT 199 or ABT 737. Rucaparib, perhaps a less well-known PARP inhibitor, one I'm going to describe to you today. And some of these newer drugs here, in fact an older drug, and drugs that have been in the clinic for quite a long time but are not well understood. So if we go back to this and we try to think for each cancer or each cancer cell what is actually required, there are a number of ways of thinking about that. And I thought one useful way might be to put a circus plot underneath it. And for any students in the audience who are interested and not aware, this site includes very good descriptions of why circus plots are a good way to image or visualise your data. But circus plots and cancer cells can become very complicated. And the idea here is that you have to envisage, particularly for these two, which we've been told enable all the rest. And I'm sorry I don't have a beautifully designed circus plot to show this. This one is one stolen from 23andMe. There are ways in which you can get from one to the other, and the way in which each cancer cell has done this stochastically and in response to whatever pressure it has encountered is the reality of what we need to deal with in providing relevant treatment targeted to that cancer cell. So how does DNA repair change over time? We know cancer incidence rises with age. Elizabeth Blackburn recently told us that telomere length decreases with age. Or perhaps more relevant for us, people with long telomeres either live a long time or have a high risk of developing a rare cancer at a young age. And that's a really interesting phenomenon that I think we're going to be hearing more about. Another way of looking at this from the point of view of the cancer cell itself is if you look at a normal cell, there is a homeostatic threshold of DNA damage and repair. And in this case, the damage is repaired. This results in predominantly healthy DNA with a low mutation rate. And any cell that actually breaks outside this is highly likely to undergo apoptosis or at least long-term senescence. But then all of our organs in our body have precancerous stages and precancerous cells which are a risk to us, but most of the time our bodies take care of that. So as you get through this period here, you can see that red is rising, so this is the cancer cell growth, and green is rising, and this is expression of oncogenes, induction of cellular proliferation, and escape from the protective mechanisms of apoptosis, autophagy, or senescence. But this rise is very slow. And in fact, what has happened is that we have these protective mechanisms, tumour suppressor genes, induction of mitotic catastrophe, induction of apoptosis, autophagy, or senescence. So all of these protective mechanisms shown in blue are really working overtime most of the time in most of our cells. And the fact that most people don't develop more than one cancer, including people who are cured of a cancer, really shows how good a job our body cells are at this. However, once a cancer cell becomes cancerous with the ability to invade, it's reached a critical stage in which these red and green are winning, particularly the cancer cell growth. And that means you have oncogenes, proliferation, and escape from these important mechanisms. Associated with this is an increase in this error-prone repair, and I'm going to show you some cartoons about this later. By now, this high driver mutation rate is winning, whereas here the body protective mechanisms were winning. The very interesting conundrum or challenge for us is to ensure that when we bring DNA damaging therapy into the mix, that we're able to circumvent the red and the green curves and indeed induce again expression of tumour suppressors, mitotic catastrophe, apoptosis, autophagy, or senescence. But the risk is that our therapies also further increase this error-prone rate of repair and this high driver mutation rate. So it is a balance between getting this right and putting the cancer cells in a position where they can be of further risk to us. So very simply, genomic stability, which is the enabling characteristic we're talking about, is dependent on faithful DNA repair and faithful chromosome segregation during cell division. And if you walk down this side of the slide, you can see what happens most of the time. So genome repair occurs because our cells incur damage all the time. We go through a G1 to S transition. If the repair has occurred properly, mitosis ensues, there are checkpoints here, spindle checkpoints here, and if the whole telomerase system and spindle assembly system is accurate, then a final pass will occur here and the daughter cells will come out being with relatively normal DNA, give or take a few SNPs, and 2N. However, on the other side, you can see the concept of today's talk, genomic instability, which occurs in a variety of ways. You can have a mistake at any one of these red spots. Basically, there are centrosome effects, spindle assembly checkpoints, further checkpoints down here that don't pick up DNA which has been accumulated. Now, if at this point the unrepaired DNA is able to trigger p53-dependent apoptosis, then that cell will die. And again, that likely happens all the time. But cancer cells are very good at evading this apoptosis mechanism, and then you have a real problem because you have a cell that has a lot of these capabilities, and at that point, it's under its own control. Now, normal cells have redundant repair mechanisms which underpins the success of most of our cells most of the time. These are an array of insults which you'd be used to seeing: radiation, chemotherapy, reactive oxygen species or ROS, replication errors just occurring every day, mispaired bases, spontaneous mutations. Now, our bodies can deal with that a lot of the time using machinery that I'm going to show again and again during this talk. The methods that have evolved to deal with these are varied for very good reason. And at the end of all of this, you have different types of outcomes involving repair, cell cycle arrest, senescence, apoptosis, and all of these resulting in prevention of carcinogenesis. But as Elise Cohn showed so beautifully at ASCO this year, and knowing as she does the love of my institution of mice, she was very willing to give me these slides. So DNA repair really is two-edged. It is a predator and a prey. So DNA repair is a protector, but we're going to make it a target. So again, put more simply, these are the DNA repair mechanisms. It is complex and interconnected, but if you break it down, you can think of double-strand breaks and single-strand breaks. If we start over here for a moment, the most simple breaks are single-stranded, and we've evolved a whole variety of ways of dealing with these depending what the error is. And these pathways are variably important in different types of cancers and different types of susceptibilities. But the real elephant in the room is double-strand break repair. There are a number of ways of dealing with this. Two are not so good, although non-homologous end joining is incredibly important in our bodies to generate immune diversity. So therefore, we actually want to have a less than competent sister who's doesn't have such great fidelity, but generates an immune spectrum that's of great use to us. But homologous recombination in bright green is really what saves us most of the time. This is high fidelity and is the focus of much of the efforts at producing new DNA repair aimed treatments. So sensing obviously becomes important because this is the way in which we're going to deal with defects. So if there is a double-stranded break, there is the break itself and then replication stress that ensues. So all the way down, we're going to look across and look at some of these key molecules. So Parp itself, we're going to talk about Q70, Q80, which are really important in a process called non-homologous end joining, and RPA, which detects replication stress. So these feed into very important constructs, such as the MRN complex involved in double-stranded break repair. You can see here ATM, ATR, these recognise damage, but they talk very specifically to molecules involved in the signal propagation, check 1, check 2, and the MDM2 P53 axis. So we have P53 again at the top here, a whole lot of cell cycle machinery, and then down the bottom, through the cell cycle, you can see what the important types of DNA repair are. Now, non-homologous end joining will get involved any old time as long as the pathway's intact. And whilst that's useful for CRISPR, it's not necessarily useful all the time if you've got a tricky precancerous cell. Homologous recombination is much better, and we do rely on that. And here you can see if there is failure of repair, a cell should undergo senescence or apoptosis. If the repair is faithful, then cell cycle progression will be allowed. If there has been misrepair, this will result in either a mutation, mitotic catastrophe, or other similar negative event, and then again, we're in trouble. So if we just look at hypoxia as one example of an everyday stress. So acute hypoxia results in replication stress and chromatin changes. This is just designed to show you the same culprits, the same partners in crime, if you like. So when it works, they're good guys, but equally, if they're co-opted by a cancer cell, they become bad guys. So again, you're seeing the same processes mentioned again and again. How does this play out over time, like I was alluding to earlier? So minutes, hours, and days and weeks along here. You can see that these initial first responders are involved, but then further on you have more complex responses and ultimately over weeks, potentially epigenetic silencing of critical processes. So if a cell is under extreme hypoxia for a period of time, many cells will die, but if they've adapted ways of getting around these control mechanisms, the cell will live and change. And this is just one example. If you look just at hypoxia, you can see the involvement of responses in all of these pathways at various levels. So given the complexity of this, and this is just for one type of insult, you can see how much work has been done and how much work still needs to be done to understand these processes in a critical way. So just very briefly, a few words about p53. It's critical in preventing cancer. The p53 pathway is mutated in many cancers. Can we actually target p53 itself? This has been thought to be too difficult up until recently, but there certainly are some mechanisms coming through. This is a nice sort of rheostat or way of thinking about it. So you have p53 upstream of the p53 effectors. You either get cell cycle arrest or apoptosis, but this is balanced very much by pro-survival genes, which can be co-opted as oncogenes. So p53, as you know, has many different functions, and you could also replace telomerase with that. But here you can see that p53, and this is our very latest data from Annie Janich and Andreas Strasser, again at the Centenary Symposium, we have well understood, or at least players that we've been working on for many years, some newer ones involving other pathways, and then very new ones involving pathways as yet unknown. So in order to understand what p53 is doing in a particular tumour cell, it's necessary to really understand the expression of these relevant effector genes. So if we want to activate wild-type p53, there are a variety of different ways that are being explored at the moment, including reactivators that can actually bind in the right pocket and restore a conformation defect. But I'm going to go back to the cell cycle now. So again, the concept of p53, it's keeper of this G1S transition. We know mutation abrogates the ability to stop cell cycling. Is there a way we can get at this in a p53-dependent fashion? So WEHI is the protector of the G2M progression to allow subsequent mitosis. WEHI inhibition removes this inhibition of mitosis, which should occur if the cell has not repaired properly. So this further compounds DNA injury and could be used to result in mitotic catastrophe, particularly of a cancer cell. So this is the first report of a specific trial looking at p53 mutant high-grade serous ovarian cancer, where there is loss of p53 in nearly all cases. So to repair damaged DNA, these p53 mutant tumours are more dependent on the G2M cell cycle, and this is regulated by the WEHI kinase. So this specific inhibitor is predicted to sensitise p53 mutant ovarian cancers to genotoxic agents. And this is just some early data presented at ASCO this year of this WEHI inhibitor delaying progression-free survival compared with placebo. So this is the active drug and this is placebo in p53 mutant ovarian cancer. This just provides a set of non-responders and responders to allow further translation to better understand this. So for the second part of my talk, I'm going to be talking about familial cancers and with a real focus on the Parp inhibitor drugs that I've mentioned at the beginning. So what have we learned from familial cancers? When we look at all cancer predisposition genes occurring in families, this provides a roll call of genes with really critical impact on cancer development. Many of these are involved in DNA repair. And that's shown on this slide with hereditary breast and ovarian cancer here, with these players that you're about to hear a whole lot more about. Mismatch repair in colon cancer and the Li-Fraumeni syndrome involving p53 itself down the bottom. So it's been 20 years during which we have chased BRCA1, it's actually 21 years, BRCA2, and perhaps this was not better highlighted than by Angelina Jolie in the last few years. So again, going back to the slide I showed before, double-strand breaks, single-strand breaks, HR, this is where the action is for BRCA1 and BRCA2. So in fact, Fanconi anemia is something we've known about for decades, and clinically this was thought of as being a syndrome that affected blood production. But what we now understand is that there's probably this subclinical Fanconi phenotype, and that these are the same genes that are involved in DNA repair and hereditary familial syndromes. So if you look, there is a whole pack of them, but they are leading the pack of HR genes, so genes involved in the homologous recombination pathway. But here you can see that these really constitute the very major genes. BRCA2 is FANCD1, BRCA1 is FANCS. Two genes that are becoming much more relevant are PALB2 and RAD51C. They're FANCN and FANCO. So these Fanconi anemia genes provide an example of the critical genes of the homologous recombination pathway. And this is just one example, and I'm not showing many detailed examples at all through this talk, but this is one example where much detailed work has come together to really provide a firm basis for understanding the reasons why BRCA1 and BRCA2 are so important in producing these cancers. And these very specific relationships, for example, between this coil domain here on BRCA1 and a coil domain on PALB2 are important. And then you have interaction with this WD40 domain, again with BRCA2, linking these three, and to RAD51 paralogs. The reason this becomes important is because so far, after 20 years of research, we've managed to define some crucial, indisputable roles for BRCA1 and some for BRCA2, and this is what we understand at the moment for BRCA1. So it's important in DNA end resection, and I'm going to come back to that in the end. It's also important for loading RAD51 onto single-stranded DNA. This creates an essential intermediate without which DNA repair by homologous recombination can't proceed. So the Parp inhibitors are the class of DNA repair inhibitor with the best track record so far. They're really leading the way. They're becoming the poster child, just as imatinib and trastuzumab were for growth factor inhibition. So this class of DNA repair inhibitor started out with a study of what at that stage was thought to be a truly rare cancer, and it was thought unlikely that we'd be able to find enough cases to complete this work. That was 2007, and FDA approval was achieved at the end of last year, which I guess shows that it does take a while even when you've got a really good story. So what is PARP? So polyadenosine diphosphoribose polymerase really provides a scaffold. We know it better as PARP cleaved during apoptosis for some of our experiments, but it's actually the scaffolding role that's most important. So you see DNA damage occurring here. PARP directly binds to single-strand DNA breaks. Once bound, it produces large amounts of scaffold to enable repair enzymes to come in and repair the DNA, after which the PARP is released. So it has long been studied for more than 30 years, back in the 60s. That's about 45 years now, I guess. So it's involved in a lot of these processes that we've touched on today, and that for each of which we have quite reasonable understanding for. It's important to point out that it, like many crucial molecules, it does different things in dividing versus quiescent cells. So in dividing cells, it's all about this chromosome integrity and spindle function. But in quiescent cells, there are many more functions involving proteasomal degradation, polyase synthesis, and transcriptional regulation. It's important to know that even though we're concentrating on the DNA repair effects, there are these other transcriptional modulatory effects, and that PARP inhibitors have been studied in inflammatory contexts for many years, perhaps not successfully because they haven't come through, but now with increasing knowledge, it is likely that we'll start to see more of this. Now, just switching to ovarian cancer as a model system, there are many types of ovarian cancer, and what we work on in my lab is this high-grade serous cancer type, which accounts for most of the type 2 or high-grade types. Most of these low-grade types are not lethal, although some are. For high-grade serous cancer, we know that around 50% of these cases have an altered DNA repair pathway for homologous recombination. This is from TCGA, and this fact alone makes ovarian cancer a very useful setting in which to study DNA repair inhibitors. This is just a slide from Liz Swisher's lab that shows, when you look at consecutive cases of high-grade serous cancer, the types of genes, most of which are in that HRD or Fanconi pathway, that are found. And if you look at the prognosis of the cases with those gene changes, so mutations in HR genes, we see the same improved outcome as TCGA reported for BRCA1 and BRCA2. And if you exclude the germline BRCA1 and BRCA2, you still see this effect. So it's likely that it's occurring for these other genes in the pathway. And it's not just a prognostic effect on its own. There is data suggesting that this pertains specifically to platinum sensitivity. So PARP has a central role in DNA repair. It can directly impact, on sensing for BRCA1 functions. And if you inhibit PARP, not only do you impact these, but you also block non-homologous end joining. That becomes very important in a moment. But the bottom line is a DNA repair defect in a cancer cell and PARP inhibition lead to DNA damage overload, which should lead to the cancer cell death. So that's the concept of synthetic lethality, and this was the very first clinical trial of a systemic therapy that was targeted to an inherited defect in patients who had the inherited defect. So this was the first trial of BRCA1 and BRCA2 mutant cases or patients with ovarian cancer, and they all received the PARP inhibitor olaparib. This was not randomised, they were consecutive cohorts, but you can see that the maximum tolerated or the optimal dose, 400 milligrams twice a day, these patients did better than patients receiving a lower dose. This was followed up in this really pivotal study. We've published this a number of times in New England Journal, and then with this update of the BRCA1 or 2 mutant subset in Lancet Oncology last year, showing an impressive improvement in progression-free survival in this maintenance setting. So these patients received chemotherapy back here and then were randomised to either the PARP inhibitor or to placebo. This improvement was retained for first and second subsequent therapies. So this was really, there was something very significant going on here. But let's look just a little more closely at these HRD genes. So what we know is that if these HRD genes in the pathway are intact, the cell will be proficient in homologous recombination leading to cell survival. But if in fact there's an abnormality in one of these critical players, then the cell will be deficient in HR and that cell should die. So if we wanted to pick which patients will benefit from PARP inhibitor therapy, the data I showed you so far is for BRCA1, BRCA2, the two best known genes, mutations that are definitely pathogenic, either in the germline or the tumour. But what about these other genes? I've listed 2 here that I mentioned before, but there are many more. And of course, there are many other ways of silencing or impacting on gene function. So what about all of this blue space? Now, we don't really, we can go and sequence all these genes, but we don't actually know which ones of those are important. And here, if you look at knocking down these genes in a range of cells, cell lines, the cell lines, of course, have to be competent for HR to start with, or there's no point knocking an HRD gene down if it's already HR defective. But then you can argue you're knocking down these genes in a context that isn't commensurate with what's happening in human biology. So there's no real great way of doing that and knowing for sure. So this was another way, first published by Abgovich in 2012. So he derived a score based on large sections of loss of heterozygosity in the genome. I'll show that a bit more in a moment, but what I just wanted to point out here is that you have the blue genes are HRD scores in BRCA1 or 2 mutant cancers, and these are the wild types. And you can see that you can pick up these wild type cancers that have an HRD score like the mutant cases. So this is important because we want to be able to pick up these red guys down this end. So this is the first genomic scarring method that has now been published with efficacy data this year at ASCO, which I'm a co-author on. And this looks at the PARP inhibitor rucaparib, which is the PARP inhibitor we study in my lab. And you're looking for these large areas of loss of heterozygosity, so the subwhole chromosome, but it's the overall percent of the genome involved. So here you have BRCA1 or 2 mutant cases. These are wild-type cases, and these are other wild-type cases. And what you can see is that there are some wild-type cases that look like the mutant cases and some that don't. So what Clovis did was take TCGA and Australian Ovarian Cancer Study data sets to devise an optimal cutoff in the setting of high-grade serous cancer. And there they were to show that if you did use this cutoff, the high genomic LOH patients had this same improvement in outcome that I showed you for the TCGA dataset and for Liz Swisher's dataset. So that meant that we were nominally dividing the population into biomarker negative, BRCA-like, and BRCA mutant, according to this genomic LOH cutoff. So this is the Ariel 2 clinical trial, which we've been running here, and this is the outcome. So These patients received single-agent rucaparib. These patients had a BRCA1 or 2 mutation in either their germline or their tumour and did very well. These patients here were biomarker negative, and these patients here in the middle did better than these. You might think that difference isn't so great. The idea here is that we need to better understand what constitutes that advantage and how to make more of it. If you like, how to make these red guys look like the blue ones. That's exactly what my lab does. You may know Monique Top, who started her PhD in setting up the patient-derived xenograft system in my lab. And we use this to study cancers from women. We define their clinically most important prognostic phenotype at the time of first having the PDX grow in mice. We look at the response to platinum, and we can clearly see that some are very sensitive, some are sensitive but only for a short time, and some are absolutely refractory. We further categorised and published these, and I presented this last year in this forum, to show that if you look at whether there's a BRCA1 or 2 defect or not, and you look at platinum response, you can see that, as you'd expect, the BRCA1 or 2 like do better, and the ones that are not BRCA1 or 2 like do worse, and actually have oncogenes overexpressed with which we're very familiar. So this is looking at the same thing for PARP inhibitor response, and if you look at a range of BRCA1 or 2 mutant PDX, you can see some respond very well, some respond somewhat, and one doesn't respond at all. And likewise, if you look at BRCA1 and 2 wild-type PDX, most don't respond at all, but one does. So what underlies this difference? So if you go back to the old accepted mechanism for PARP inhibitor efficacy, DNA damage would create a single strand break. PARP inhibitors were known to block base excision repair. So then when a replication fork was encountered, a double strand break would ensue. So with a double strand break, this would normally be repaired by error-free repair, homologous recombination, but in a BRCA1 or 2 deficient cell, error-prone repair mechanisms were required. They weren't very good at their job, and double-strand breaks wouldn't be repaired properly, and you would end up with a whole lot of genomic instability and cell death. Now, that theory is still quoted in many, many publications, but if you drill down and understand the defect, the detail, that is leading us astray. So Scott Kaufman showed beautifully that you could knock down PARP1 and get a response to a PARP inhibitor, veliparib, and this is in an ovarian cancer cell line, pair of cell lines, PO1s and PO4s. So PO1s lack BRCA2, PO4s, the BRCA2 mutation has been complemented and it's back in frame. But if you actually knock down this critical base excision repair pathway member, XRCC1, you don't see this effect at all. So the student in his lab proposed that it wasn't that simple and that in fact we needed to look at non-homologous end joining. He then went on to prove the point that the NHEJ pathway is actually required for the genomic instability induced by PARP inhibitors, and that if you don't have an intact NHEJ pathway, you won't see efficacy to the PARP inhibitor. So what does this mean and why does it matter? So if we've already said that the PARP inhibitor actually drives NHEJ and that that's what leads to cell death, then clearly if NHEJ is missing, like down here where Q70 is missing, that means that loss of NHEJ will result in drug failure. That means that we can now know that we need to go looking at the NHEJ pathway or other pathways or genes impacting upstream of that pathway that do something similar. And at the end, I'm going to come back and give you an example of this. So if you don't have NHEJ, you don't get genomic instability, the cell survives, and you end up with drug failure. So if you look at resistance to PARP inhibitor therapy, it's an array of mechanisms of drug resistance that are well known to you in other settings. There are a couple of really interesting examples that are specific for this though. So Liz Swisher and Alan Ashworth both showed in 2008 that under the pressure of PARP inhibitor, these mutations could revert. So as you know, that means you've got a whole lot of tumour cells that are behaving stochastically. If one randomly develops a change that puts that mutation back in frame, that cell will be selected for. And you might have killed all the other cells, but that is the clone that will grow through. Liz Swisher went on to prove that that was the case in human cancers. And if we take that as an analogy, mutation reversion, you can see that you can have actually a wider range of DNA repair pathway reversions, and I've just described NHEJ loss as one of those. That BRCA1 or 2 mutant PDX that didn't respond has a particular mutation that is susceptible to structural reversion. And then if you look, you could see oncogene-driven mechanisms, drug efflux, and then environmental influences can also be involved. And we have examples of nearly each one of these in our PDX. I'm just going to mention 2 today. We have some interesting reversion mutants that we're studying, and an ongoing collaboration with Scott Kaufman looking at NHEJ. I just wanted to come back to this. So I made the point before that BRCA1 was critical for DNA end resection at the end of HR and for loading RAD51 onto single-strand double DNA. So this is a beautiful paper just published using an siRNA screen looking at ways of escaping high-dose olaparib, and the 2 top hits of that screen were Rev7 and its upstream partner, 53BP1. And what this shows is that the NHEJ hypothesis is almost certainly correct, and loss of Rev7, which occurs during treatment on a Parp inhibitor, can result in HR competence and PARP resistance. So although I don't have time to go through the detail of that, it's really fascinating to watch these discoveries come through. So we described PARP inhibitor development, so this is Liz Swisher and Scott Kaufman, my 2 collaborators, and we particularly looked at issues that went wrong during the development of PARP inhibitors in the hope that the next drug or cab off the rank might not have to be slowed down on a number of occasions by discoveries that weren't in fact correct. I just wanted to mention the concept of super responders, although I don't have time to go through the detail, and this is emerging. This similar concept is being described for platinum responders, BRAF inhibitor responders, and has the potential to tell us a huge amount. I just wanted one slide on toxicity because it's such a beautiful mouse model. So one of the most worrying toxicities for PARP inhibitors is the emergence of erythroid hypoplasia, aplastic anemia, myelodysplastic syndromes, and potentially acute leukemias. Now let's put this in context. All our DNA damaging chemotherapy does this too. But is there something special about PARP inhibitors that we should be aware of so that we can manage it better? So this is a beautiful paper, again a recent one, and it shows that it's loss of PARP2 but not loss of PARP1 that is responsible for this. A series of beautiful experiments that really underpin what we're seeing in patients. We only see it rarely, but when you do see it, it's very scary on behalf of the patient. We now have PARP inhibitors in first line following ovarian cancer first line treatment, only in clinical trial setting, not in the clinic, but still, we need this sort of information to help us manage. So should we develop PARP1 specific inhibitors? Probably not. All of the best inhibitors at the moment are PARP1 and PARP2 inhibitors, but at least we could potentially develop protective strategies or management processes. So I said I'd talk about beyond ovarian cancer, and there is a lot of work going on in these other cancer types. 2 very interesting stories, pancreatic, BRCA1 and 2 mutations are not common, but where they occur in this very devastating cancer type, we're seeing some really neat responses. Gastric cancer, this was studied in ATM deficient gastric cancer in Japan, and a really nice response was seen. And my final example is that of Ewing sarcoma. It's the 2nd most common cancer in children, but that still makes it a very rare cancer. So in a screen using NCI cell lines, it was noted that Ewing sarcoma had very high levels of PARP1, an associated gene, and low levels of HRD proteins when they went to understand the basis of this. They developed a gem and proved that olaparib was exquisitely powerful in combination with other standard forms of chemotherapy, and this has now resulted in a combination clinical trial. So just one thought about designing combination therapies. If we did have a correct Circus plot and looked at all of these interactions, I could already show you examples in preclinical models and clinical trials for nearly all of the potential combination therapies that you see in front of you, some more compelling than others. So it's not an easy space, but this is what we need to do. I don't think that single agent therapy will cure many cancers other than perhaps those really critical super responder cases, although even there, the super responders are best occurring as a maintenance following DNA damaging chemotherapy. That also gives us the clue that sequencing treatments might be the answer. So if you give one treatment, then another, depending on the evolutionary changes, perhaps picked up by circulating tumour DNA, specific for that particular cancer, then we might be able to follow through with really specific sensitivity. So DNA repair inhibitors, the best class of which are the Parp inhibitors, were first understood in BRCA1 or 2 mutant ovarian cancer. This is being extended to DNA repair defective subsets of most cancer types. Really, all cancers will become rare with increasing molecular annotation, and this story really proves that by studying truly rare cancers, we can define driving biology for therapeutic exploitation and then apply this more broadly to other cancer types. So we have a lot of interest in rare cancers in Australia and at Royal Melbourne and at WEHI. I just want to finish in my last 4 minutes with a few thoughts about the immune system, exercise and diet. And this is a feel-good session about the impact on and of DNA repair and life itself. So this is an example of why clonal evolution matters in a tumour. So if you look down here, you can see the concept that tumour antigens are expressed and recognised as self at some point in the beginning, and that allows the tumour to not be wiped out by the immune system. But as the cancer progresses and genetic divergence occurs, there are increased numbers of infiltrating T cells observed with immune inhibitory receptors. Now, at some point, tumour neoantigens, so new antigens that are developed, are then recognised as non-self, and this allows the immune system to attack these particular tumour epitopes. I've already mentioned that there are multiple ways in which Parp1 impacts on inflammation, and indeed, this is an array of immune cells that are also directly impacted by Parp and susceptible for Parp inhibition effects. And this is Elise Cohn's slide from ASCO demonstrating the really novel ways in which we think we may see interactions between DNA repair inhibitor therapy and various types of immune therapies. But of course, we need to understand tumours because they're not all the same. And about 15 to 20% seem to be an easy target for immune therapy. This is a really nice review, and this shows you tumours divided into 4 groups. This set have an adaptive immune resistance, this has immunological ignorance, this has tolerance, and this has intrinsic induction. So if we actually do routinely, which we're moving towards doing, screen each cancer for their immune profile, we may well be able to start to harness this as well. And exercise, it's very hard to control exercise trials, but there is very neat data emerging showing that exercise really does enhance the immune system, and this is just some data showing that patients perhaps are able to tolerate a higher dose intensity just of their standard chemotherapy. So that means they're more likely to complete the chemotherapy that they've been prescribed. So whether this means that exercise is a positive thing overall for cancer or not, I'm not sure. But if we look at the hallmarks and instead put a few everyday activities around them and retain our focus at the bottom, what you end up with is this. You should all watch that sugar film if you haven't seen it. Healthy eating just really relies on coming back to primary produce. Not quite sure what your view on meat is. Exercise, clearly, if you're under a stressor, is not a problem. Alcohol, I don't think you can have none, but perhaps moderation. And we're way past smoking. So if you do all of these things, you should reduce the need for DNA repair, reduce the inflammatory load on your body, reduce cancer incidence, and improve cancer control. I don't speak about cure. I think control should be our aim. So these are some papers that might be helpful for the students. You have to define the difference between an apple and an apple covered in toffee. And these are the many participants in the work in my lab, both in my lab itself, our international collaborators, and very importantly, the Australian Ovarian Cancer Study. And we thank women with ovarian cancer, their families and their friends, and our particular WEHI Buddy System consumers. Thank you.
last slides, exercise and PARP inhibitors. Any sort of connection there?
No, not yet, but I think we should work on that.
So why don't you want to find a PARP1 selective compound?
Great question from a structural biologist. So we don't yet know whether that would be sufficient, and PARP2 certainly could take on the roles of PARP1 in many cases. I think one of the problems with developing new drugs is that we tend to scare away or become frightened of potential toxicities instead of learning how to manage them. And I think by understanding them better, we have a better capacity to do that. This is a very rare side effect, although it's one we want to know more about. So I think it would probably be a harder ask for a selective PARP1 inhibitor to work than to make sure that we can protect people against developing that toxicity.
You mentioned the idea of tumour profiling, like individually sequencing patients' genes. Can you just speculate on the actual costs that would have, and like whether cancer treatment in the future would it favour the rich, so to speak?
Yeah, sure. And look, at the moment there's definitely a two-tiered system, but we're struggling as fast as we can to bring that back to some sort of level playing field, particularly in Australia. So if we talk about Australia as opposed to the US, where you have different subsidy schemes involved, in Australia, the best way for you to get your tumour sequenced if it's reached a compelling stage where you've moved beyond the availability of standard of care or proven evidence-based medicine really is to enter a research project if you don't have $5,000 to pay for a foundation medicine test. But at the same time, major genomic centres in Melbourne, Sydney, Brisbane and beyond are all developing that capability. So I work closely with also the guys in Perth, and really what we'd like to see over the next 2 years is that with, for example, the rollout of the Victorian Genomics money, that's all done within research settings that can be accessed, that people know they can be accessed. There are many cancer cases that don't need that at a particular point in time, and at the moment we really have to focus that availability on compelling cases, cases that are not behaving like they should, cases that are progressing beyond reasonable treatment availability, and having medical oncologists all focused together and knowing how to access those systems is really important. So it really means that we've seen a huge meeting of the minds from the private system coming into the public system for that purpose. That's really only just beginning, but we should see a lot more of that. In the long term, what we would say at the moment, for example, is that you could probably gain reasonable information from $1,500 in the public system. Now that won't be quite as good as the gold, what I consider to be the gold standard of a foundation medicine test, which costs about $5,000, but a lot of that's in the pipeline. And if we get the bioinformatics pipelines right, we'll be able to bring that costing down.
There's two questions there. Brian, thanks.
So, carrying on from that, you're also talking about immune profiling of individuals with cancer. How much does that cost?
So that's much more straightforward. That's done by immunohistochemistry or immunofluorescence. Again, so that's been best done in melanoma, and colorectal cancer have also worked out a pretty good paradigm for it. Ovarian cancer is looking at that right at the moment, but really labs, and again, major labs in each of the capital cities are ensuring that they have a platform. Now, there are various ways of making that more complex than it probably needs to be. If you actually have a very good pathologist and you just look at activated CD3s and CD8s, whether they're in the tumour itself or in the stroma infiltrating the tumour, you can actually already come up with a pretty good score according to that 4-part profile that I showed you. So one issue is to try to actually keep that simple, but we do need to look more closely across different tumour types because that hasn't been done yet.
So you said that one way forward, going forward, might be to change the way you might schedule administering drugs to try and kind of track, the best optimal progression of trying to preempt what the tumour might do next, I suppose. Do you think there's a good way going forward to try and balance that? If you already have existing standard of care for treating a certain patient, but you think that there might be an alternative treatment strategy that may in the future be better based on some of these approaches, is there a way of balancing these two things?
I think, in fact, the example that I showed you, which was of maintenance therapy, so bringing the Parp inhibitor in after DNA damaging chemotherapy, that made sense particularly because the DNA damage would synergise with the Parp inhibitory effect. But I think just the concept of sequencing maintenance therapy after proven therapy is something that's very useful. We've used that a lot in the clinical trial space. What we're doing now, which we haven't done before, is really looking at ways of assessing, for example, with ovarian cancers, trying to ensure that they undergo a molecular screen soon after diagnosis. Now, at the moment, patients receive about 6 months' worth of chemotherapy in any case. And in order for us to give a turnaround for a reasonably complex array of molecular or novel tumour testing, which includes next generation sequencing for mutations, amplifications, and some immune profiling and other immunohistochemistry and ISH, that would probably take us around 3 to 6 months. Now, most patients will get through their treatment and not need to change tack during that time. The ones that look like they're not doing well, we could fast track their testing. If you then either look at triaging patients onto a first-line maintenance, or at least for those that relapse, streamlining them and triaging them into a relevant therapy, you can see that that kind of timeline of 6 months of novel tumour testing is workable. So I think that's the answer. We want to triage based on which hallmarks we find to be adversely affected in each cancer, and then have an array or a suite of phase 2 clinical trials. So this is all about having small, smart, nimble clinical trials and having a national infrastructure that supports that. So there are a lot of endeavours underway, both at the clinical trial groups level, at the NHMRC ethics and governance level, because that's a huge slowdown, and at the way we as academics interact with pharma. So we have 8 pharma at least that we interact closely with, and we're having more scenarios where those pharma come together and talk with us about multiple drugs, multiple trials, and understanding the concept of triaging. 2 years ago, that wasn't the case. So I do think that there's light on the horizon.
One last one.
Are Parp inhibitors given orally?
They are. I'm sorry if I didn't make that clear. They were originally given IV, but they are oral therapies. So another thing we're doing is particularly combining them with other oral therapies so that patients don't have to come in for chemotherapy. The management of toxicity is also a little bit easier because you can, be on the phone to someone at home and modulate their dose a little if you need, just to get them through the early time period, which is when you tend to see toxicities.
Thanks, Clare. Thank you.